phospholipid, and it performs all the function. This model was rejected because later came the model of the sandwich mechanism. According to the sandwich model, there were two models given by scientists. One was Danieli and Davison in 1935, and the second one was by Robertson in 1959. And according to both of these models, with some modification, both models stated that a plasma membrane is made up of 4 layers, and that 4 layers are first there will be an outer protein layer and an inner protein layer, and in between 2 layers of phospholipids are present. This model could not explain many of the properties of this plasma membrane, so it was rejected. And finally came the model which is called as the fluid mosaic model. This model was proposed by Singer and Nicholson in 1972. This model happened to explain all the properties of the cell membrane as well as all other organellic membranes. That's why so far it is the most accepted model to explain the structure and properties and the functions of any biological membrane. According to this model, phospholipids are present in the form of bilayers. So this is a single layer of phospholipid, this is the second layer of phospholipid, and the phospholipid layers are arranged in such a manner that their hydrophilic portions are facing either the outside or facing the inside. Inside means towards the cytoplasm. And the two phospholipid layers or phytol chains, these chains are facing the inside. So the outer portions are hydrophilic or polar, and the central portion is hydrophobic or non-polar. In between there are present proteins. Those proteins are basically integral proteins or peripheral proteins. So this is an integral protein region. It is present inside the membrane. This is considered as the peripheral protein. This one is considered as again a peripheral protein. This is again an intrinsic protein. This is also intrinsic, this is also intrinsic, but please remember this is also considered as a tunnel protein because it forms a tunnel from one end of the cell membrane towards the other end. So things can easily go across through this protein. So integral protein can be small in size or they can be complete, traversing from one end of the membrane to the other end. Such are called as channel proteins or tunnel proteins. So there can be proteins present on the surface. Those which are present on the surface are easy to be removed and they are called as the peripheral protein. Now the next thing is differences between the two types of proteins. Integral proteins and peripheral proteins are different, reason being, first thing is the integral proteins, they cannot be easily separated because they are present embedded inside the membrane and they are a part of the membrane. It is easy to remove the peripheral proteins because they are present just on the surface. And the second reason is why integral protein cannot be easily separated. The reason is it develops hydrophobic bonds with the phospholipid. These bonds are strong, and because these bonds are strong, it is not easy to separate these proteins from the plasma membrane. The next difference is they are embedded inside the bilayer. They are simply lying on the surface, either on the outer surface or in the inner surface. They may act as, these proteins may act as a channel protein or they may act as a carrier protein. Carrier protein is something which will pick up a molecule from one end, take a 180-degree turn and then release the content on the opposite end. So integral proteins can either be a tunnel protein or channel protein, otherwise they can act like a carrier protein. There is no such role of the transport in case of peripheral proteins. 70% of total proteins present in a membrane are integral proteins and 30% of these proteins are peripheral proteins. So that's why this model given by the Singer and Nicholson simply stated that a cell membrane, according to this model, is something like a protein icebergs which are floating, or they are always in a lateral movement, sidewise movement in a sea of phospholipids or lipids. Now what are the functions of a Functions of a cell membrane or a plasma membrane. So first thing is, the plasma membrane holds the content of the cell intact. So it somehow maintains the integrity and individuality of a cell and its components. It provides protection, it provides individuality and compartmentalization. So one cell becomes different from the surrounding because that compartmentalization is done by a selectively permeable plasma membrane. Selectively permeable means, permeability means which allow things to go across. Plasma membrane is selectively permeable because it allows certain things to go across but would not allow the other things to go across. That means it is selective in permeability. So the other function of the membrane is the recognition. Recognition means surface to surface interaction between various things. For example, a cell interacting with the environment and understanding the changes in the environment, or a cell coming in